Hi, I'm Evan Cromwell, President and CEO of Protein Fluidics, and thank you for coming to our poster, Automated 3D Cell-Based Assays Using a Novel Flowchip System and High-Content Imaging. This will be a short video where I'll go through some highlights of the data and other information in our poster. If you'd like more information, please download the poster or contact me for additional information. Great. Let me first acknowledge the co-authors and the people who helped on the poster from Protein Fluidics, Michelle Long, Anthony Tai, Rashmi Rajendra, and from Electric Devices, Oksana Sarenko, Matthew Hamer, and Carol Crittenden. And my information is there if you'd like to contact me or go to our website for more information. What we're really trying to show here is the 3D cell models, which are now becoming more and more important in drug discovery and biological research, is using automated microfluidic systems as well as high content imaging to really elucidate more information from these very precious and elegant cell models. The workflow that we've developed is based on the novel Flowchip, which I'll describe in a little bit later. And those are loaded with organoids and reagents. And then those Flowchips run your assays and all your assay protocol steps automatically inside an incubator. It's a very simple, intuitive operation where protocols are preloaded. You press play, and when the assay is done, you can take the plates out, which are in the standard SLAS ANSI footprint, and read it on any type of analysis system you can really imagine. In this case, we'll be highlighting how it can work with the high-content imaging systems. So the instrumentation, as I mentioned, is a PUMA system and its associated accessories, as well as the Image Express high-content imaging system, confocal imaging, and it's just controlled by the MedExpress software. The Flowchips, while they look like a 384-well plate, are actually a lot more than that. You have Flowchips with 8 tests and 8 lanes. Each lane is an individual test, and those are connected by microfluidic channels on the back. In the center is a sample well where you can place your organoid or your spheroid, and then the other reagent wells allows you to move fluids inside and out, in and out of those sample chambers. This video shows a little bit how the protected chamber works. So in the bottom of each well where your organoid is placed is a sample chamber about a microliter in volume, and you can then transfer reagents in and out from the side of that without disturbing your organoid. And also when you do these transfers, you don't dry out the organoid, which is very important for a lot of assay applications. And remember, these assay changes, reagent exchanges all occur within an incubator. So you're doing in situ sampling, in situ reagent exchanges with your organoids. So the two applications, the first one I wanted to go through briefly is oncology disease modeling. In this application, we're using cancer spheroids, HeLa spheroids, or HCT116 spheroids, or HepG2 spheroids. And those are placed into the well where we do a compound incubation, do an overnight treatment of them. Following the treatment, then we would add in the stain, and then we can wash the stain out if you need to do that. And once all these processes occur, and these all occur in your incubator, the plates are removed, and you can remove supernatants or conditioned media for downstream analysis, or put the plates onto your imaging system to do your high-resolution, high-content imaging. This shows some of the analysis that you can do with the Image Express microsystem and MedExpress software, where you take the organoids or the spheroids, and then you can segment the spheroid from the background and do analysis on a cell-by-cell basis within the spheroid. In this case, counting number of cells, total cells, live cells, or dead cells. If you want to look at the compound treatments, then each lane in your flow chip can be a different concentration of a compound. In this case, we're showing Staurosporin treatment for both the HeLa cells and HepG2 cells. And you see a nice concentration-dependent response of those. And if you do the image analysis, you can see the increase in dead cells concentration and decrease in live cells, and get EC50 values of this. In addition to the image analysis, as I mentioned, you can pull out the supernatants. In this case, we look for VEGF in the conditioned media from these different spheroids. We actually didn't see any VEGF in the HeLa spheroids, but in HepG2, we saw a nice decrease in VEGF expressions, VEGF secretion, and Staurosporin increase. From these, then you can measure the EC50 values for the different compounds, as I mentioned. So the second application is slightly different. This is a neurospheroid functional assay. Here we're showing the ability to do multiple compound additions and concentration increases with the flow chips. So if you imagine you have a single spheroid in a well here, You can load it with dye. You can then rinse out the dye, or you can add in media. You can then add in increasing concentrations all to the same spheroid using the automated process steps. And because it's so easy to take the plates out, you can remove the plates and measure the plates on, for example, the flipper system, and then put it back into the Puma system and continue the assay process. So it gives you a lot of flexibility in the type of assays you want to run. So in this case, we're looking at these Microbrain 3Ds from Synopsis, and really cool things. You can see them beating here or firing. As the neurons fire spontaneously, you have the calcium flux and then change in fluorescence. And then on the flipper system, you can measure those increases and decreases in fluorescence and get a firing rate, if you will, out of each neuron. So with the Puma system, then you can do either different concentrations or different compounds, as well as an increasing concentration in the same neurospheroid. So again, these are the same microbrain that was subjected to different concentrations for 30 minutes and then read on the flipper system. So it gives you a lot of flexibility and a lot of capabilities for working with these type of organoids. So in summary, I hope I showed you how we have developed an easy-to-use organoid assay system that combines with high-content imaging to give you multi-parametric information about your organoid samples. It works with a wide range of organoid samples and automates many of the protocol steps that you are using, whether it be reagent exchanges, staining, immunofluorescence staining, conditioned media sample, etc., and compatible with multiple downstream analysis, as we showed in here with high-content imaging, which image stress microsystem. So again, thank you for coming to the poster and your attention, and please download the poster and contact us for more information.